learning object. You will be able to describe the process of translation. Imagine we have a beautiful article. If we want to copy this effect, then we need some particles or object, and it will take more or less time. Similar strategies are used by cells. The most important task for a cell is protein synthesis, which based on codes in DNA. But how are these codes read? What's the process that helps decode the language? Protein synthesis merely by the sequence of A, T, G and C. The answer to all questions lies in the central dogma of life. So, what is exactly the central dogma? DNA makes a copy with itself by the process of replication. Post-replication in every cell, the code of DNA is cracked and transcribed into messenger RNA. This messenger RNA is then read and the code is translated into proteins. This interesting concept is nothing but the central dogma. We know the replication process is semi-conservative in nature. So now we will be focusing on the two processes, transcription and translation. Transcription and translation together is called gene expression. The code of gene need to transcribe first and then translated to form the protein. This protein becomes functional. For this gene expression can be divided into two processes, transcription and translation. In our previous class, we already discussed about the transcription procedure. Today, I want to discuss about the process of translation. So, dear students, let's go to the discussion. The process of polypeptide chain synthesis according to the information encoded in mRNA is known as translation. Translation is the second step in protein synthesis and it occurs in the cytoplasm. Inside the body, the process of translation occurs within every single cell. Each cell has a nucleus. After transcription, messenger RNA is moved out of the nucleus and enters the cytoplasm. This messenger RNA strand acts as a template for protein synthesis. Some enzymes are present in the cytoplasm as amino acid tRNA synthetase. The enzyme macromolecule has two binding sites. This enzyme macromolecule has two binding sites. One site recognizes amino acid, that means methionine. This is followed by the binding of the ATP molecule and release of pyrophosphate, resulting in activation of amino acid. Finally, the transfer RNA and the activated amino acid bind together. This amino acylated transfer RNA is known as methionine transfer RNA and released from the enzyme. This work started at the first stage of protein synthesis, initiation stage. During this stage, a small subunit of ribosome binds to the messenger RNA strand. This messenger RNA strand is made of codons, which is sequences of 3 bases. Then the ribosome subunit moves along the messenger RNA in 5 to 3' direction until the recognizing AUG or the initiation codon. At this point, methionine tRNA possessing the anticodon UAC base with AUG codon of the messenger RNA. Then the large subunit of ribosome combined with the small subunit of ribosome. The large subunit shows three sites: amino acyl or A site, peptide site or P site, the exit site or E site. This all unit form the initiation complex. This followed by the elongation stage. Elongation stage. During this stage, another transfer RNA carrying molecules of amino acid process the messenger RNA ribosome complex and fix in A site. Then a bond is formed between methionine and another amino acid molecules on the transfer RNA. As a result, methionine tRNA becomes deacylated. The ribosome then advances a distance of 1 codon and deacylated transfer RNA shifts to the E site. From a dissociates meanwhile, another transfer RNA carrying amino acid molecules attaches to the A sites. This followed by the binding of the amino acid molecules. Repetition of this process leads to the formation of amino acid chain. This event is called elongation. Finally, when UAG codon or stop codon reaches the A site, elongation is terminated. Termination is the last stage of protein synthesis. The chain of amino acid molecules is released from the ribosome. This released amino acid chain is the protein, and this part of protein synthesis is known as translation. This tRNA detaches from the messenger RNA. Then ribosome also detaches and disassociates in large and small subunit. Homework. Write down the difference between transcription and translation.